Using CRISPR Cas9 to edit gene expressions in crops. Introduction and driving question. The ability to change a plant's genes determines how well we can improve crops, affecting things like growth speed, nutrition, and disease resistance. For centuries, humans relied on slow, traditional breeding, but now we use modern, molecular tools like CRISPR. Therefore, the question remains, can we effectively use CRISPR to target and change specific genes in crops to benefit us? Traditional methods are slow and often occur by chance, while CRISPR-Cas9 allows for fast, exact cuts in DNA. Understanding these differences is key to seeing how technology can improve global food supplies. This experiment explores the molecular process of CRISPR-Cas9 and compares its efficiency in disabling genes to the traditional breeding methods used in the past. Background research. What is CRISPR? The gene editing system CRISPR-Cas9 was invented by bacteria serving as a natural defense against viruses. Even though CRISPR-Cas9 was not an invention of humans in the late 1980s, scientists began to learn how to manipulate the bacterial defense system to benefit mankind. Today, with CRISPR-Cas9, scientists can enhance human health and develop disease-resistant crops. They continue to use it, furthering advances in all aspects of scientific research from agriculture to medicine. Essentially, the CRISPR-Cas9 system is like the immune system of bacteria. When a virus infects a bacterium, it collects a small component of the virus's genetic code and stores this information in a particular region of its DNA called CRISPR, a clustered regularly interspaced short palindromic repeats. Essentially, this is like a database of passing counters with particular viruses. When the particular virus strikes again, the bacterium uses this early acquired information to produce another component called the RNA guide in association with the Cas enzyme. The guide component tells the Cas enzyme to search for similar segments of the virus's DNA in a cell, and when located, the Cas enzymes cut the DNA into small segments, thereby inhibiting its replication. This system of bacterial defense has been utilized by scientists as a precise tool for gene editing. They can design their own RNA guide in which specifies the Cas enzyme to cut the target area in the organism's genetic code. And this has a wide range of applications, such as the destruction of a dysfunctional gene, replacement of a normal gene or a set of healthy genes, and an addition of a completely fresh gene or set of genes. This was one of the biggest breakthroughs in science that occurred between the years 2010 and 2012, as scientists such as Jennifer Doudna and Feng Zhang were able to develop a protocol for using CRISPR in mammalian cells. This breakthrough opened a wide range of opportunities in the prevention of treatment of genetic diseases, the development of quality agricultural products such as crops, as well as understanding the function of genes. The CRISPR technologies are being extensively investigated in laboratories all over the world, and they aim to unlock novel therapies for the treatment of cancers, plants with increased ability to resist the effects of global warming, and exploring means for fixing long-standing genetic disorders. Yet, despite the vast possibilities afforded by the development of novel biotechnology, there are plenty of areas that still merit rigorous scientific study, especially with regards to issues like previously predicted adverse effects and so-called off-target editing. Researchers are being very prudent so that they can correctly assess this remarkable resource that has been developed by nature for the past 1 billion years. Key vocabulary for understanding CRISPR: CRISPR, Cas9, spacer, base pair, guide RNA, genome, gene editing, tracer RNA, PAM, double-stranded break, off-target effects, HDR, nucleotide, eukaryotic cells, viral DNA, gene expression, mutation, non-coding DNA, and template DNA. What is STEM SOS? What is STEM education? Why is it important, and how can it shape the future? STEM is an acronym for the fields of science, technology, engineering, and math. Discussions of STEM-related programs has become a presidential priority because too few colleges students are pursuing degrees in these fields. STEM-based education teaches children more than just science or mathematical concepts. They focus on hands-on learning and real-world application, helping develop a variety of skill sets, including creativity and 21st century skills. As workplaces become more automated, capabilities that include creativity and complex problem solving become more critical than manual skills or memorized content. STEM education must prepare our workforce to innovate and work with modern technologies, and also to consider their societal effects. U.S. Department of Education. What is STEM? STEM Education Coalition. STEM SOS model. Project-based learning, PBL, is a constructivist instructional approach where students are engaged in a meaningful inquiry of personal interest to them, and where collaboration and personalized learning are emphasized. Research confirms that PBL is an effective and enjoyable way to learn and develop deeper learning competencies required for a successful college, career, and civic life. Buck Institution of Education. STEM SOS is a regular interdisciplinary standards-focused and engaging STEM teaching approach that is teacher-facilitated, student-centered, and directed through sets of project and inquiry-based PBL projects. STEM SOS PBL approach is to maintain the focus on standard-based teaching while enriching and understanding the learning of students through PBL projects. The goal is to promote not only collaborative skills and student ownership of learning, but also to promote student success in state and national standards. STEM SOS PBL Showcase is designed to promote and share exemplary student work and PBL projects that can serve as valuable learning and teaching tools for students, parents, teachers, and other educators. Zain, your turn.
Did you pause?
Nah, it doesn't need to be paused.
So for the MyPBL page, from disease resistance to enhanced nutritional value to improve crop yields, the ability to modify plant genetics is critical to advancing our agriculture productivity and food security. Agricultural methods for genetic improvement have evolved dramatically over time, ranging from traditional selective breeding that requires years of cross-pollination and cultivation to modern gene editing technologies that can make precise changes to DNA in a single generation. This difference in approach affects not only time required to achieve desired traits, but also precision and predictability of the results. Past our introduction, our driving question is, can we effectively use CRISPR to target and change genetic specific genes in crops to benefit us? And some variables we used are our viral DNA sequences, our guide RNA and template DNA strands. And the methods we used are going to be DNA sequencing, microbial gene research, pattern identification, viral DNA comparison, viral invasion, foreign DNA identification, and many more. We use different gene editing methods such as method 1, gene disruption, method 2, gene replacement using HDR. And some of our materials are going to be, we have 2 sets of materials, reagents and supplies, and biological materials that are stored at negative 20 degrees Celsius. And we do have our regular lab equipment as well as our safety equipment such as disposable laboratory gloves, UV safety goggles, and hot gloves or heat-resistant gloves, and some safety. And then we have our safety procedures, which is our general laboratory safety, such as wear disposable laboratory gloves and UV safety goggles at all times as good laboratory practices. For our procedure, we did module 1, designing gRNA to target SH2, and then we did module 2, just to determine gRNA specificity. Then we did module 3, which is digesting with CRISPR, which is our laboratory work. Then we did module 4, Argos gel electrophoresis. Then we did module 5, which was staining with flash blue. This is after we got our results and visualized them. And afterwards, we have some photographs of us working on the project with our experiment and running it. And our results that we found out is that the gel was stained with flash blue, so DNA appears to be light blue. And this is our and we found that the gRNA successfully guided Cas9 to cut the SH2 genes, and gRNA B also shows multiple bands, which is similar to gRNA, and this shows that gRNA B successfully cut into the SH2 gene, and the fragmented DNA bands in each single well shows that the DNA was successfully cut. And now we use the BLAST specificity results and the gel. The results directly support the BLAST analysis, as gRNA B and C were highly specific to the maize SH2 gene and successfully cut the DNA. Now for application and further research, we found that we could use this technology in modern agriculture and addressing food security challenges beyond creating super sweet corn by targeting SH2 gene. And this experiment opens up avenues for further research into gene editing technology and its optimization. We have many different references, as you can see on our website, and we have a research paper after that. For our social studies page, we dived into the background of CRISPR. What is CRISPR? How is CRISPR used? And what CRISPR does and how it's used specifically towards crops, and how it edits the crop genes for more safer and better crops. For our ELA component, we did a journal, and basically this journal documented our every step through this experiment, such as from week 1, from finding out what project we're gonna do, what group we're gonna be in, as well as the rest of our steps, such as conducting this experiment and when we got our results. Thank you for your time.